Hello, my name is Jason Breuler, and today we'll be talking about the detection of cyanobacteria in formalin-fixed paraffin-embedded tissue, otherwise known as FFPE tissue. Before we begin, I would like to thank my co-authors, Wasim Selwanes and Andrew Kleinhenz, as well as my faculty mentors, Dr. David Kennedy and Dr. Stephen Haller, for their immense help with this project. Now you may be thinking, what is cyanobacteria? Cyanobacteria are microscopic organisms found in many water sources. They are also referred to as blue-green algae and are the main organism present in harmful algal blooms, otherwise known as HABs. These bacteria are able to produce many different toxins, such as hepatotoxins, neurotoxins, cytotoxins, dermatoxins, and other irritants. One notable toxin produced is microcystin that can cause long or short-term damage when highly concentrated, leading to a variety of different diseases. Taking this background into mind, this project looks at testing a protocol for the extraction and purification of cyanobacteria in FFPE tissue. The purpose of finding a reliable protocol is to be able to study the effects of these cyanotoxins in organ tissue that has been preserved in these FFPE tissue blocks. Specifically, this project looks to get the best purity index values for a small sample of FFPE tissue to contribute to the successful extraction of DNA. Before cutting or running any samples, each FFPE block sampled to was assigned a 5-digit identifier in order to autonomize the human tissue that is used. Once autonomized, multiple 10 micrometer samples were cut using a microtome. To extract and purify this DNA, the GeneJet FFPE DNA extraction and purification kit was used as a control. Some highlights of this protocol were a 1.5-hour water bath incubation period, along with an additional 40-minute heat block incubation to remove all paraffin from the sliced samples. Additionally, the extraction and purification process involved multiple rounds of centrifugation and the addition of different solutions, such as RNase A binding buffer and ethanol, in order to make sure that the cells are fully broken apart and prepared for column filtration. Once prepared, 2 wash buffers were used before extracting the DNA with a elution buffer. Using this method, the results were analyzed for duplicate samples of 4 FFPE blocks, making 8 total. 3 of these were healthy tissue, and 1 was carcinogenic tissue. A 260/280 ratio was used to express the sample purity, with a value of 1.8 indicating that the sample was generally pure. Additionally, the nucleic acid concentration and nanograms per microliter was measured using nanospectroscopy. For the purpose of FFPE tissue, a value at or above 100 nanograms per microliter was the target for good extraction. Looking at the data from all samples collected, it was concluded that the GeneJet FFPE DNA extraction and purification kit showed good performance in extracting the DNA samples. The purity of all samples was within an acceptable range of 1.7 to 1.9 for the 260/280 absorbance rating. Additionally, a majority of the concentration values were above 100 nanograms per microliter, except for one healthy sample labeled 12584 that showed values in the 70 range. Taking this data into consideration, the main goal of this research was accomplished to optimize a method for extracting and purifying DNA from FFPE tissue samples in order to obtain high-quality DNA for analysis. Over this past semester, many additional samples from a variety of disease states and target organs have been collected and are currently being processed with this optimized protocol. These samples will be used to measure presence of cyanobacteria in order to determine whether a correlation exists between disease severity and cyanobacteria presence. Thank you for your interest in this project, and I hope that you were able to learn a little more about the extraction and purification of DNA today.